Okay, everyone, this is going to be a cool overview of mitosis. But before we get started, I just want to remind you, take your time with the video, number one. Pause it when you need to. Take notes when you need to. Write questions down when we need to, right? Because that way you'll have your questions ready for when we go over it in our FaceTime or our Google Meet time. All right, so some of this stuff we've talked about before, so just to understand, the cell cycle is different from mitosis. Cell cycle, when we talk about the cell cycle, that's the complete life of the cell from beginning to end. We know we have three phases for the cell cycle. You can have interphase, which is the beginning phase where it's doing its normal job. It then grows and duplicates and gets prepared to go into mitosis. Then you can have mitosis, which is actually the division of the nucleus. And then you have cytokinesis, right? Those are your three phases of mitosis. Now, you can also look at that with meiosis, right? Interphase, meiosis, and cytokinesis, and the categories kind of follow the same trend, okay? So now that we're going to talk about specifically mitosis, even though we're specifically talking about mitosis, we need to start it with interphase, but let's just talk about some general things about mitosis. We know mitosis is needed for growth and repair. So again, like we've talked about before, you can't have anything grow without mitosis, or the reproduction of cells. You can't have any cells or any organs or any parts of the body be repaired without mitosis, right? It's only in your body cells, or what we call somatic cells. So that's things like hair cells, skin cells, bone cells, right? And we talked about earlier the few cells that do not go through mitosis, and you might want to go back to your notes and double check which cells do not go through mitosis. And then we know mitosis basically has four phases. So prophase, metaphase, anaphase, and telophase. These are the only four phases of mitosis. Now I know in some other videos you saw things like pro-metaphase. Those are just transitional phases, right? So pro-metaphase is the in-between of prophase and metaphase. But when we're talking for right now, I want you to focus on four main phases: prophase, metaphase, anaphase, and telophase, right? You should be able to know what happens in each phase and draw a diagram to represent that. And we're going to go through that in this video today. So when we get to diagramming, we know interphase is not part of mitosis, but we need to talk about it. It's the preparation phase, right, where the cell is doing its normal job. In interphase, you cannot see chromosomes. So if you look at this diagram right here, you cannot see individual chromosomes. In this phase, they're in big clumps together, and we call it chromatin, right? That's that darkened area of the nucleolus that's full of chromatin. But chromatin is just a bunch of chromosomes grouped together, and you cannot see the individual chromosomes. In interphase, and we'll talk about this a little bit later, more specifically, we know the cell is growing and duplicating. And when we say duplicating, we mean it's doubling its number of chromosomes, right? It's making duplicate copies of the chromosomes. Also for interphase, the nuclear membrane is still intact and still complete, and that's very important. All right. Going on to prophase. We know prophase is the first phase of mitosis, right? This is what the diagram will look like. Key points. First phase of mitosis. This is the first time, excuse me, that we can see individual chromosomes. So these X-shaped things inside the nucleus, those are our individual chromosomes. Also in prophase, the cell membrane is going to start to dissolve. Now, you have to remember, this is a phase, right? So you can have early prophase or late prophase. Early prophase, you might still see a lot of the cell membrane, the nuclear membrane. Late prophase, you might not see any of the nuclear membrane. But we know somewhere in prophase, that nuclear membrane is starting to dissolve. Why is that dissolving of the nuclear membrane important? Because of what happens in metaphase, or what has to happen in metaphase. Metaphase is our second phase of mitosis. These T-shaped things, what we call centrioles, those start moving towards the poles. And when we say poles, we just mean the opposite ends of the cell, right? So there's one centriole on one, what we'll call the north pole, and another centriole on the south pole. These excuse me. These centrioles produce things called spindle fibers. Spindle fibers are going to come out of the centriole and attach to the centromere, which is the middle of the chromosome, right? Now, in metaphase, all of these chromosomes are going to line up in the middle of the cell. The only way this can happen with chromosomes lining up in the middle of the cell is for the nuclear membrane to be gone, which we know had to happen in prophase, right? So again, everything happens for a reason. Nuclear membrane dissolves and gets disappeared, so the chromosomes can come out of the nucleus and line up across the middle of the cell. Again, this is a process, right? So early in metaphase, you might see spindle fibers starting to come out of the centrioles. Late in metaphase, at some point in metaphase, more towards the end, these spindle fibers are going to go all the way and attach to the centromere, right? So again, it's a process, right? They'll slowly come out and eventually attach to the centromere, which is the middle of the chromosome, right? Why do they attach there? Again, everything happens for a reason. Something has to happen in the next phase. So we know our third phase of mitosis is anaphase. That's where the chromosomes are split together in half. How did that happen? The spindle fibers attach to the centromere, which is in the middle of the chromosome, and they're going to contract or tighten. And when they tighten or pull away, right, they're going to split those chromosomes in half and create chromatids, which you should know by now is half of a chromosome, right? So you're going to have the chromatids moving to the poles because these spindle fibers are going to pull them back towards the poles. So this group is going to get pulled back towards the North Pole. This group is going to get pulled back towards the South Pole, right? So they're going to pull back away from each other to the poles. Fourth and final stage of mitosis, telophase. A couple of key points for telophase. The chromatids are going to arrive at the poles, right? Then you see this dotted line showing back up. That is the nuclear membrane starting to redevelop because these have to go back into the nucleus. In order for us to have a nucleus, we have to have a nuclear membrane. So this nuclear membrane is going to redevelop. Right here you see the pinch in the cell membrane. Why? Because these are going to start separating into two distinct cells. So it's going to pinch the cell membrane. Now, if you remember, plant cells is going to look a little bit different. Plant cells do not have just a cell membrane. They also have a cell wall, which is very rigid and very stiff. Because the plant cells have that cell wall, which is rigid and stiff, there cannot be a pinch. So what happens is the cell plate, this dividing line across the middle, shows up, and that's where the new cell is going to form. So if you look at it, you'll have one cell on top here and one cell on the bottom. So in plant cells, we're going to have a cell plate. All right. And that is pretty much the end of mitosis, our 4 phases. Once mitosis is over, of course, we stroll into cytokinesis. It's not part of mitosis, but we still have to talk about it. Cytokinesis, right? The division. We have 2 new cells and they're exact copies of the original cell. All right. So that's just the diagramming for mitosis. Keynote. So understanding what happens on the inside, right? So you should know that, remember we said in interphase, the cell is going to duplicate itself, right? So if you look at, let's take this top example. In the beginning of interphase, I'm going to have 4 chromosomes. That 4 represents chromosomes. 4 chromosomes on the inside. If it duplicates itself, by the end of interphase, it doubles itself, I'm going to have 8 chromosomes. All right, again, this is the end of interphase. That cell is going to go through mitosis, prophase, metaphase, anaphase, and telophase, which we know is the splitting of the nucleus and the dividing of the chromosomes, and it's going to give me 2 exact copies. Again, 4 and 4. If it splits it, right, 8 split in half, 4 and 4. So if you notice, what I had in the beginning, 4 chromosomes, I'm going to have 2 with 4 chromosomes in the end. All cells that go through mitosis is going to go through that same process. If I had another cell that started off with 23 in the beginning of interphase, it will go through interphase, duplicate itself. By the end of the interphase, it'll have 46. Go through mitosis, split itself, and now I have 2 cells with 23. So when you look at it, it doubles and splits once, so I'll have the same thing in the end, two of the same thing in the end that I started with. It's kind of like if I have $5 and I double it to $10 and then I split it in half, I'll have two sets of $5. Same concept, right? So again, I hope this was helpful. Write down any questions that you have, rewatch the video if you need to, pause it, take notes, right? And then when we meet, we'll go over and get your questions answered and clarified.